Hello, my name is Yanbi. Together with my co-chair, Julia McNabb-Balta, welcome to the third session of this morning. So we're going to be talking about pancreas development and the lineage of plasticity, relevance to exocrine pancreas disease. So this will be a great session. We have three amazing speakers. So the first speaker is Dr. Steve Pendle. He's the director of basic and translational pancreas research group and professor of medicine at the Cedars-Sinai Medicine. He actually has contributed about more than a few decades of career to basic science in pancreatology and also pushing significantly the translational property of basic science to clinical practice. Today, he will discuss about the exocrine pancreas microenvironment and diabetes in PDAC. Thank you, Dr. Pendle.
Thank you so much. How's that? Hey, there's a button you got to push here. All right, I'll start over again. But anyway, thank you for attending. So my talk is going to be taking off a little bit from what you heard already this morning. In fact, some of my slides are actually the same as some of the slides you've already seen. I just didn't know what's going to be presented. But I'll try to make those brief and really get to the bottom line here. So what I'm going to talk about is the impact of the pancreas microenvironment on the development of PDAC, focusing on what's going on in the islet cells. So the talk is going to be divided into a couple different segments. First is going to be epidemiological associations, which actually is a little bit of repeat, and I'm going to go through those slides pretty fast. Then beta cell function in patients with pancreatic cancer. Actually, Phil talked about that a little bit earlier. But then finally, I'm going to focus then on islet cell alterations in pancreatic cancer patients with diabetes or without diabetes. And then the use of this information for potential therapy using animal models. So this is actually what Suresh showed from a paper from their group, showing that, and just to set the stage again, is that in pancreatic cancer patients, most of the patients, he said almost all, but it looks like most of the patients have some form of either diabetes or glucose dysglycemia compared to a control population. So that shows the importance of diabetes association with pancreatic cancer. The other study that I wanted to present was showing that the survival is not so good in patients who have pancreatic cancer with diabetes. And it's mainly, in this study, also important to recognize it's a meta-analysis showing in long-term diabetes in the top part of the graphic compared to those with more short-term diabetes, that in the top graph, long-term diabetes associated with poorer survival in a variety of studies shown in this forest plot. It was less clear in patients with short duration of diabetes before the diagnosis. And that's the bottom one. So the next part is, again, a little bit of a review of what I think Phil Hart presented, and that is beta cell dysfunction in patients with pancreatic cancer. And I'm going to start, though, with a study that was done a little bit before what Phil presented, and it's a study from colleagues of mine from China, from Nanjing. And it was studying the secretory characteristics of beta cells in patients with these different groups. And as you can see in the dotted line, that's the glucose levels, insulin levels in patients who were given a meal tolerance test. So at 0 time, they were given boost. And then insulin levels were measured over time, and the time is in minutes in the horizontal axis. And you can see in the dotted line a normal response. In type 2 diabetes, which is the upper line, you can see in type 2 diabetes there's actually an overshoot in the insulin secretion, which we would expect. However, in the two bottom lines, one with normal glucose and the other with pancreatic cancer and diabetes, the first one with pancreatic cancer and no diabetes, you can see in both groups there was an inhibition of insulin secretion compared to both controls and in patients with type 2 diabetes. That was an initial finding that actually was important setting the stage for understanding that, as Phil pointed out, diabetes and pancreas cancer is different than it is with type 2 diabetes. The next study is actually that Phil presented, which was recently published, shows, and I'll just repeat it very briefly, that insulin secretion is markedly inhibited in patients with pancreatic cancer and diabetes compared to patients with type 2 diabetes. And I think the middle, on the left, the middle graphic shows that the best, where with patients with type 2 diabetes in the red line, with a meal stimulation, there's significant insulin secretion, but in patients in the blue line with pancreatic cancer, there was inhibition of secretion. And the bottom graph is just a validation with C-peptide measurement that he presented. And then also, as he presented, there's an overshoot in glucagon. On the left side is the glucagon response we expect to see in patients with type 2 diabetes, and compared to control, with type 2 diabetes in the open circles on the left, you see that there's some increase or not inhibition of glucagon secretion with patients with type 2 compared to control population. But on the right side, you see in blue that even compared to type 2 diabetes, where the glucagon secretion is increased, it's even greater in pancreatic cancer. So we have two major findings. One is that insulin secretion is inhibited, and the other is glucagon secretion is exacerbated in patients with pancreatic cancer. I think those are critical findings, and I think lead to a better understanding. So then the next part that I'll focus a little bit more on now is that can we identify what the disorder is in the islets? And so we used different techniques in patients' tissues that were operated on where the tumors were removed. And we divided the patients into patients who had diabetes compared to those who did not have diabetes. And so that's what this data is going to be about. And we use two kinds of methods for investigation. One is spatial molecular profiles, which measures RNAs in tissues in specific areas we can identify, so-called regions of interest, ROIs. And the other is imaging mass cytometry, where we use antibodies to identify certain signals that identify cell types and even signals and constituents of those cell types. And again, in all these measurements, we used samples that were stored in the biobank from our patients who had pancreatic cancer resections. So the first piece of data is really to show how we identify the islets, whether they're stained with insulin, you can see on the left in green, or glucagon in red, purple. And what we use is those antibodies to identify the cell type. So obviously insulin would be more of a beta cell type and glucagon would be more of an alpha cell type. But the interesting finding, just with these antibody measurement, you can see on the non-diabetic patients, you see a lot of green. Those are the insulin cells. In the diabetic patients, the green is basically absent and replaced by glucagon. That actually goes along with the whole person studies that I described from both China and the United States. So that was very, very important for us that we had something that made sense. Okay, so the next slides are going to try to detail some of the molecular signals that are associated with this. And so I'm going to start in this slide in the left-hand side. And what we're doing here is we're measuring RNAs, and see the dotted line at 1. Those are what normal levels would be. And you can see that we have in blue, we have non-diabetics, and in red, we have diabetics. And so starting on the left-hand side, you see insulin, INS. You can see in both diabetics and non-diabetics, the insulin amount in the islets was significantly decreased. The IAPP is also a beta cell marker, and it's going along with the decreased insulin secretion. The next thing I want you to point on, I want you to see is where the circled indicators are, and you see GCG, that's glucagon, and then next to that is TTR, which is a marker in alpha cells. And you can see that in both cases, there's some increase in the non-diabetics, but in the diabetics, there's a marked increase. So that signal, this is an RNA signal, corresponds and validates the antibody signal that I showed earlier. The other key signals I want to point out are XBP1 and DDT IT3. So what are those? Those are actually the focus of our study, so I need to explain them a little bit. So those are ER stress signals. In secretory cells, both in the exocrine pancreas and endocrine pancreas, XBP1 is necessary for both the formation of the cell into a secretory cell and necessary for the functional operation of those cells, secretory cells. DDIT3 is also referred to as CHOP. It's kind of another ER stress signal that's often associated with cell death. And you can see that both of those are the XBP1 is decreased, and the CHOP or DDIT3 is increased in both the patients with diabetes and non-diabetes. And then finally, I'll just point out that pancreatic polypeptide on the furthest right was extremely increased, and Phil Hart pointed out that they're decreased in the blood in patients with pancreatic cancer, and I said, oh my God, so there's something wrong with the secretory response, but the RNA is there. So I don't know what that means yet, but that's an interesting finding. Okay, so then this is just a representation of the glucagon expression insulin to glucagon expression in the tumor islets, and the non-diabetic on the left and on the diabetic on the right. And you can see that there's a significant marked change in the ratio. And again, insulin decreased and glucagon increased. Okay, I wanted to show you just a cartoon about ER stress signals, and then I'm going to come back to how that might apply. So ER stress pathways have generally been described as initiated by misfolded proteins. They get stuck in the ER and they set off these signals. And, but those signals can be changed by environmental factors. And I'm going to show you one later on, interferon gamma, that can change those signals as well. But the point here being is that, that you can see on the left-hand side in the green, I guess I have two greens, but on the green, that's RRE1, that's leads to the formation of this ultimate transcription factor called XBP1 that's processed, and it's important again for the functional secretory capacity of a cell like the beta cell or the acinar cell. We study this a lot in acinar cells. On the right, you can see the signal starting with PERK, and it leads to this increase in CHOP or DDIT3. And you can see that can lead to cell death or an adaptive response. And I think we're going to see there might be an adaptive response. Okay, so to get back to some of our measurements, this one is the IMC. This is where we use antibodies and identify different cell types and their signals. And again, this was just looking at how we could look at a variety of different this slide is just how we can look at a variety of cell types in the tissue. And you could see it's kind of complicated. There's a lot of different cell types, and they're scattered. But the point is that these can be measured with techniques that are available to measure these kinds of signals in the tissue. And so this one just shows insulin, glucagon, macrophages, both M2 and total macrophages, just to show you the complexity of the tissue. And you can see again on the left there's more insulin than on the right, which is the non-diabetic on the left, and on the right diabetic where there's less insulin. Okay. So in order to analyze the data, I have to go back to kind of understanding pathways and then see if the data fits. That's how I set up a hypothesis here. So the hypothesis for the pathways involved in the effect of PDAC on the microenvironment on islets goes like this in the hypothesis. That is that, and this has been mentioned earlier, that cancer cells through different signals like TGF-beta, for example, PDGF, FGF, can have an effect on cancer fibroblasts. And those cancer fibroblasts then actually can feed back to the cancer, but in this graphic I'm just going forward, they can secrete a cytokine called CXCL12. CXCL12 is a very interesting signal in cancer research and pancreatic cancer, and interacts with CXCR4 receptors on both cancer cells, but also macrophages and T cells. And in the literature, I have references below, the literature would suggest that one of the outputs of CXCR4 containing macrophages and T cells are interferon gamma, IL-1 beta, and TNF alpha. So So is there evidence? Oh, and these, the other point is, by references, is that these are known to affect insulin, insulin processing and secretion in beta cells. And maybe even the inhibition of glucose regulation of glucagon secretion. Okay, having said that, are these signals in our tissue? So this is a display of data from the analysis, and starting with left, showing the islets, and then on the right, the islet vicinity. And so what we looked for here is T cells that are CXCR4 positive, T cells CXCR4 positive, whether they express CXCR4 or not. Remember, that's a signal that responds to CXCL12. And you can see in the diabetic patients, it was much greater, significantly greater than in the non-diabetic. So we have one validation of this hypothesis we presented. And that's also true in the islet vicinity. So those are the cells in the microenvironment around the islet that you can see. Again, there's a difference in the lymphoid cells, total in the left part of the right-hand side, and T cell CD4 specific. And you can see in both cases, the diabetic patients have a greater amount. Then we asked the next signal down the line. Remember, those cells can secrete interferon gamma. So the next signal down the line was interferon gamma. And this graphic shows the expression of interferon gamma in islets in the cancer cell departments in the diabetic versus the non-diabetic patients. And so on the left graphic are the insulin compartment, on the right, the cytokeratin department. That's the cancer compartment around the islets. And you can see in both cases, the diabetic patients have more measurement of interferon gamma compared to the non-diabetic patients. So again, another confirmation or piece of evidence to support the hypothesis that interferon gamma is involved. We did try to measure IL-6 and IL-1 beta, which were two other cytokines I mentioned. We didn't see a difference. So interferon gamma seems to be a focus of at least the data that we're coming up with. So going back here, just to reiterate, we demonstrated that, there is CXCR4 positive cells, and that those CXCR4 positive cells can secrete interferon gamma. And from the literature, we know on the furthest right that we know from papers that interferon gamma inhibits the XBP1 and increases the CHOP. Those are the two ER stress signals I talked about that we identified in the tissue. And that those are associated with decreased insulin production and increased glucagon production. So that's our current evidence, our data. We continue to work on the data. These bioinformatic, or what do you call it? These data files are enormous, and you keep finding things the more you look. Okay. And so the final thing is, well, does this have relevance to maybe a treatment? So we have a project with Sanford Burnham group here in San Diego, actually, on trying to determine if we could change the beta cell signaling so that it affects the cancer. And so with our colleagues here at Sanford Burnham, we have animal models that have CHOP ablation in the beta cells. And we can measure metastasis of cancer, and that's what I'm going to show here, but we also can measure the effect on the primary tumor. And the point being is that in the bottom left, under A, you can see that in the bar graph, you can see that when we have the beta cell CHOP deletion, we actually had a marked inhibition of metastatic lesions when we give cancer cells into the liver of animals. That was dramatic. And we also see effects in the primary tumor as well. And in this particular case, we used genetic ablation, but we also have delivery of siRNA to the beta cells attached to GLP-1. And when we do that, we get the same result. So we can get more specific delivery of CHOP inhibition to the beta cell with an siRNA. So these point out that maybe this ER stress pathology that leads to the changes in insulin glucagon is important in the development of, or in the promotion is a better word here, promotion of pancreatic cancer both locally and metastatically. So the key points, in summary, are that pancreatic cancer diabetes is unique and associated with decreased insulin secretion and augmented glucagon secretion, which was already presented. But what we show is there are marked alterations in insulin glucagon expression in the pancreatic islets, and there's a presence of an inflammatory immune microenvironment, and we can show that there's activation of endoplasmic reticulum stress, and that if we can decrease that, we change the outcome in animal models, at least, of pancreatic cancer. And so the bottom line and the bottom statement is that we think that development of treatments that deal with the pathophysiology we were defining here may be more advantageous than current treatment with insulin, which actually insulin may be deleterious, and studies show that because the cancer cells actually have receptors for insulin which promote its growth. So I want to acknowledge this was multidisciplinary and multi-site investigation. And I showed you the Chinese study initially from Dr. Bao and Dr. Li in the upper right, left-hand corner. The study that Phil showed with Fred Toledo, Phil Hart, and Mark Godarzi in the center across 3 institutions in the United States. And then the islet-specific studies with Richard Lugea, Richard Waldron, Aurelie Lugea, and Janine Hamede, all within our group. Janine is a medical student, and Richard and Aurelie have been working with me for many years now. And then from Sanford Burnham, Jing Yang and Randy Kaufman, who provided the animals and actually are intimately involved with all the analysis that I've described. And these are our funders. You can see a lot of different funding agencies. And the Consortium for the Study of Chronic Pancreatitis, Diabetes, and Pancreatic Cancer, which is a generation that we talked about here from a number of studies, a number of centers that actually were conceptualized by Dana Anderson and helped us carry those out in the United States. So thank you very much, and I'm finished.
Thank you so much for this wonderful presentation, Dr. Pendle. We're going to be holding the question at the end of the session. So our next presenter is Dr. Farzad Esni. He is Associate Professor of Surgery at the University of Pittsburgh. His focus is on crosstalk between endocrine and exocrine compartment in the pancreatic regeneration and cancer. And he will present today on acinar to beta cell conversion.
Thank you. So which one should I? This one. Okay. Thank you very much. I would like for the opportunity to present our data. I'm going to switch gear. Okay, there it is. And talk about diabetes a little bit and intersection between acinar cells and islet cells. So just the disclosure, I'm just the inventor of this. So this is a rodent islet, and as you see, we have these functional beta cells in the core of the islet, and then it's surrounded in the periphery by the other endocrine cells like alpha cells and somatostatin producing cells and so forth. In a type 1 diabetes, because of autoimmunity, and this is of course it's a very, very simplified version of the disease. So basically, many of these, if not all, beta cells are attacked and destroyed. And then in type 2, you start to have a lot of non-functional beta cells. You still have lots of beta cells, but many of them are non-functional. And during course of disease, you may actually lose beta cells as well. So the bottom line is that you're losing, in both cases, for different reasons, of course, you're losing the number of functional beta cells. So throughout the years, there has been, investigation, or attempt or efforts to find new sources of beta cells. And I'm not order is not of importance or anything, just mentioning the work that has been done by many investigators. Islet transplantation, trying to promote proliferation of pre-existing beta cells, or new genesis. It could be from ES cells or adult stem cells, or converting non-pancreatic cells to insulin-producing cells, or converting pancreatic cells like ductal, acinar, or other endocrine cells into beta-like cells. So what we have done is, we used inhibition of focal adhesion kinase to see whether we can convert acinar cells into insulin-producing cells. And so what is FAK, or focal adhesion kinase? It's basically, it's a non-receptor tyrosine kinase, and it mediates, as ECM signaling through integrins. And the downstream pathways and, the targets, basically, it's everything. So it's not that helpful when you study that. So you can find anything you want. In the context of pancreas, it has been shown that, in the developing pancreas, it's involved in epithelial branching. And that, if you inhibit the function of FAK in human pluripotent stem cells, it favors endocrine differentiation. So a couple of years ago, we published this paper. We knocked out conditionally FAK in the developing pancreas, and we saw that it was involved in timing of acinar differentiation. But what was interesting was that if you look at the to the left there, so we find clusters of cells that co-express insulin and amylase, which, as you know, it never happens. So, as you know, during development, we have the endocrine, we have the common pancreatic progenitors, and then we have the endocrine and exocrine. And these two lineages are totally separate from each other, and you don't find acinar cells it's expressing beta cell markers, especially in the adult. So. So this could be, because just an abnormal pancreatic development, because we knocked out this gene in the developing pancreas. It could be that, we have found cells that are beta cells transitioning into acinar cells, or what we would like to see, these are acinar cells converting into beta cells. So as I said, this was a genetic approach. So we found this, this available FAK inhibitor. There are a number of those available, and most of them in a cancer setting. So we treated the mice. These are wild-type mice, and we saw that it didn't have any adverse effect on endocrine or acinar or exocrine function. But what we saw that, and these are wild-type mice, we saw that these weird structures or cells that transforming into insulin or into islet cells based on morphology or stuff like that. So, but that wasn't conclusive, of course. So we turned into the reporter line. We targeted the tomato expression into acinar cells, and then treated the mice with the drug. And the idea was that if based on lineage tracing, if we see red cells expressing insulin, that means that we have converted acinar cells. And that was exactly what we saw. So when we treated the fecal, we didn't see anything in the islets, any red cells. The red cells were specifically in acinar cells, but within islets we saw some. These islets, these cells, we call them ADIP, acinar-derived insulin-producing cells. They, infiltrated the islets, as you see, and they lost amylase expression. And also they express GLU2, which is a marker for mature beta cells, functioning beta cells. They also express PDX1. And when we quantified, we saw that, basically 40% of islets contained some tomato-positive cells. And then, totally when we calculated, the contribution of these acinar-derived insulin-producing cells was less than 2%. So then, are they functional? So we treated the mice, and these are again, wild-type mice with low-dose STZ. So low-dose STZ streptozotocin, so it kills the beta cells, but it's sort of not all of them if you give low dose. So, and we saw half of the cohort, they didn't reach that 300 threshold, and they recovered on their own. And then the remaining, they passed that, and then they never recovered. And that's important because then when we treated the mice, next round, so we treated them with STZ low dose, and then we picked only those that were above 300 because we knew that they would never spontaneously recover. And we gave the drug. That's a blue line, and we saw that they started to decline at the blood glucose. Improvements we saw, and then the red, which is just STZ and vehicle treated, they didn't. And so we repeated that experiment in a ELA tomato mice, the reporter line. And what we saw was that, after STZ, again, we picked the cohort that, have higher level of blood glucose above 300. At that time point, we gave this FACI treatment for 3 weeks. When we harvest the pancreas, just a week after that, we saw that, we have infiltrating cells, but they are still not expressing insulin. I don't know where the pointer is. Is it this one? Or not this? This one, yeah. Yeah, much better. I should have asked it before. So we see that, we have infiltrating cells, but they do not express insulin. However, a little bit later, 2 weeks later, we saw that, the infiltrating cells, some of them express insulin. But that was low dose. So how about high dose? So that basically kills all beta cells. So then you have to put them on insulin because otherwise they will die. So we gave the high dose STZ, we put them insulin pellet and started the treatment for 3 weeks. We monitored these mice, and we saw that after a couple of 10 weeks after, we saw that here it is. So the STZ plus FACI treated mice, they had improved, partially improved, not 100%, but partially improved GTT. And this is a normal pancreas. This is just FACI plus saline. And this is just STZ, you don't see much of beta cells, the red signal, and then we see increase again following FACI treatment. And quantifications show that we regained roughly 25% of beta cell, original beta cell mass, which is significant because in diabetes, you can lose 75% of, up to 75% of your beta cells and still not be sort of diabetic. You might have issues, but you're not considered as diabetic. So we did that in monkeys, or NHPs, non-human primates. Basically repeat the same experiment, but for a longer time. So we induced STZ induced diabetes, and then we put them for a 3 weeks regimen, and then that's it. And then followed the NHPs. And what was interesting was that when we saw that with a significant drop in the dependence on the exogenous insulin requirement. Again, we saw around 25% recovery of original beta cell mass, or beta cell area in this case. Sorry. Now, of course, as you know, we have to do these fancy transcriptomics. So we did that. What was interesting and very surprising was that we found, of course, there are heterogeneity among acinar cells. That's well known. But what we saw two clusters of acinar cells, acinar 1 and 2. What was interesting was that this acinar 2 was what would we expect, normal acinar cells. Whereas these acinar 1, they were hybrids. They had, acinar features, but also endocrine signature. And when we treated mice, we saw that, some of them, a bridge was formed here towards beta cells. And also our trajectory showed that, these cells actually can convert to beta cells upon treatment. So to validate that, we just looked at wild-type mice. We did insulin, glucagon, and amylase. So insulin in green, amylase, and glucagon in red, and then amylase. And I've done it, since I was a grad student. I never paid attention to the amylase staining that much. But with that in mind, I looked at more carefully, or not, I didn't do that. The people in the lab did. And we found that, there are actually cells, acinar cells, with low or basically no detectable level of amylase in vicinity of the islets. And EM showed the same thing. These are acinar zymogens, and then there are beta granules there. And we also did it with RNA scope. And normally, in the tele-islet area, where basically normal, where the exocrine cells are, you don't see any insulin mRNA. Or in the proximity of the islets, you see, or in the islet, peri-islet area. Now, we did this ELA tomato mice used again. We treated them with the inhibitor, and we saw that the peri-islet acinar cell vectors start to express insulin. This is a confocal staining. So this is sort of a follow-up paper that we're working on. We have digged more into this bioinformatics, and we found that actually there are 3 different acinar cells here. And to make it easier, so we call them these are just regular acinar cells, and then these hybrid cells, we have 2 different hybrid cells, acinar beta hybrid cells. But interestingly, we also found, acinar hybrid cells for every endocrine lineage. So it's not for beta cells. So for every endocrine lineage, we have an acinar, hybrid. And these are all in the peri-islet area. Actually, I'm just working on a newly discovered biorender, so this is just apologize for the quality. But so these are acinar, normal acinar cells. I've highlighted here the peri-islet acinar cells. The pinkish ones are just normal acinar. I just wanted to highlight the area. But within them, we have these hybrid cells that they're residing in that niche. So what's the purpose of this? So we looked at different conditions. Maybe there's a spontaneous conversion. So we know that beta cell proliferation declines by age, and it's not that much to start with, but even that little amount declines as we get older. So we thought maybe there is spontaneous conversion in older mice. So these are just ELA tomato mice just treated with tamoxifen, nothing else. So in 6 months we don't see any conversion, nothing. And then at 9 months we started to see some cells here and there, but at 12 months we saw cells, residing within the islets that are expressing insulin, and these are red cells, indicating that there is a conversion spontaneously. When we quantified, we saw that the number of islets containing tomato-positive cells increased, and then the number of tomato-positive cells per 120 islets also increased, of course, and there was a variation. Most of the islets, had less than 5 tomato-positive cells, and a few of them, had, a little more than that. So we think that this indicates that by the time, beta cell proliferation normally declines, this as a backup mechanism kicks in, and to maintain a normal, number of beta cells. So in summary, so we have identified these hybrid cells, and as I said, we have acinar endocrine hybrid cells for each lineage, and they are mainly prominently found in the peri-islet area. They might also be, scattered throughout the acinar tissue. It's hard to find, so I don't know. I'm not excluding that. These hybrid cells might spontaneously convert to each respective endocrine lineage. We know that age is a factor. This happens, and what did I show is that we see also converted acinar cells to other endocrine cells. Glucose alpha cells, somatostatins. Like pancreatic polypeptide we couldn't find because the numbers are too low. So we're still working on that. And then whenever we do, in a setting of context of inhibition of focal adhesion kinase, we tap into a pre-existing mechanism. So basically we just accelerate something that sometimes, in some cases, might happen. And this acknowledgement. So Purvius and the current members of the lab, and Mohamed Saleh, he's an endocrinologist, work with us on this project. It's been a collaboration, within institute in the country and outside in Europe, and these are the funding agencies. Thank you.
Thank you so much. Our next speaker is Dr. Kathleen Del Giorno. She is assistant professor from Cell and Developmental Biology from Vanderbilt University. She will discuss endocrine cells formation in the PDAC.
Okay, got it. All right, thank you very much, and thank you to the organizers for giving me the opportunity to share our work today. So, as everyone in this audience knows, pancreatic cancer is a terrible disease, and this is largely due to late diagnosis. So by the time the cancer is diagnosed, it's likely to have already metastasized, and this leads to the patient being ineligible for surgery with a tumor that's recalcitrant to therapy. And so early diagnostics are really reliant on us having a better understanding of early events in disease progression, and so my laboratory has a major focus on this process of metaplasia. Metaplasia is the transdifferentiation of one cell type for another, and it's often considered to be a developmental reversion due to re-expression of factors associated with the development of that organ. And so a common example of this is Barrett's esophagus, that I think a lot of people are familiar with, or the idea that in response to injury, the esophagus takes on a phenotype similar to the intestine. Now, in the pancreas, both chronic injury and oncogenic mutations in KRAS, the most commonly mutated gene in pancreatic cancer, lead to Asner to ductal metaplasia. And at the most basic level, metaplasia is the transdifferentiation of these Asner cells, which have a dedicated role in producing digestive enzymes, into ductal cells. So during this process, they lose their identity, they stop making digestive enzymes, they become ductal cells, and it's thought that if the injury does not persist, or if it's resolved, that this ADM is lost and the pancreas can be populated by normal Asner cells again, at least in murine models. But in the presence of an oncogenic mutation, ADM persists and progresses to neoplasia, and with tumor suppressor mutations, then cancer. A lot of recent studies have shown that people without diseases of the exocrine pancreas harbor both metaplasia and neoplasia, and that most of them do not progress. And so it's really important for future studies to determine what causes these low-grade pannin that supposedly everyone has to move on to cancer. Okay, so a couple years ago now, we asked the question of what cell types or states result from this process of Asner to ductal metaplasia. And to answer this question, we used genetically engineered mouse models of lineage tracing, where we labeled normal adult Asner cells in mice with yellow fluorescent protein, and then induced chronic injury with cerulean, which is essentially an Asner cell toxin that causes replacement of these healthy normal Asner cells with these metaplastic ducts, which increase with treatment over time, as well as infiltration of immune cells and fibrosis. So we induced metaplasia, we performed FACS to collect these YFP-positive cells, and then we did single-cell RNA sequencing. And as compared to the normal pancreas, where you would only see Asner cells, we're now seeing multiple populations forming in response to injury, including several secretory adult cell types that are not found in the normal pancreas, but are found throughout the GI tract, including tough cells, for example. And we've previously shown that these cells will form in the pancreas in response to injury or oncogenic mutation. We've shown that these are tough cells by now canonical markers such as PAO2F3, and then also by electron microscopy, which identifies cells with the characteristic structure of these microvilli. These cells are understudied. They've been shown to mitigate helminth parasite infection in the intestine. So the question of why they would form in a de novo gland in the pancreas has been something that's been interesting for the field. This is relevant to human disease. They also form in ADM associated with human chronic pancreatitis. So as compared to nearby normal, when we identify metaplastic lesions, we can identify tough cells here by a marker called phosphogurdin, but we've identified them using other markers as well. And in single nucleus RNA sequencing studies of chronic pancreatitis, it's been published that tough cells are a de novo cluster that form, and they also express a number of the genes associated with murine tough cells, including PAO2F3. In this study, we also identified a cluster of mucus-producing cells, which is not a surprise for an injury-associated gland, but we found that there's enrichment of gastric, particularly pyloric markers in this population as well, several of which are highlighted here. And again, we validated that this population is absent from the normal pancreas, but forms in metaplasia in response to chronic injury, where you can see here by immunofluorescence, and also by electron microscopy, where we now see these population of cells with these zymogen granules that are admixed digestive enzyme and mucin, very similar to what forms in the injured stomach. So what's important to note at this point is that all of our research so far suggests that these populations are functional. So this is not an accident of injury or cell state switching. We are seeing the formation of these secretory cells which secrete factors that appear to, thus far, suppress disease progression. So in a previous study, we showed that tough cells form in response to oncogenic KRAS mutation, and when we delete them, keep pushing the wrong button, here we saw a more rapid progression of disease to cancer through modulation of the stroma. More recently, we put out a study where we're looking at these pyloric markers that we see come on during metaplasia, and we found that these cell types secrete glycans that actually inhibit disease progression. So this was recently accepted. Okay, so now for the focus of this talk, I'm going to talk about enteroendocrine cell formation during metaplasia. So again, this is immunofluorescence of lineage tracing. So we show here that this YFP in the normal mouse pancreas is restricted to amylase-positive acinar cells. It's not expressed in chromogranin A-positive islets, but in response to chronic injury, we now see cells that co-express both chromogranin A and YFP, showing that they are derived from acinar cells. When we looked more closely at our single-cell RNA sequencing, we found this somewhat concerted program of enteroendocrine cell formation where we identified an NGN3-positive precursor population, as well as ghrelin-expressing epsilon cells, serotonin-expressing enterochromaffin cells, pancreatic polypeptide-expressing gamma cells, and somatostatin-expressing delta cells. And although pancreatic polypeptide and somatostatin are expressed in islets, we did not see insulin or glucagon. We did see minor expression of other hormones like gastrin and CCK, suggesting that these cells are more similar to enteroendocrine cells found throughout the gut. So thus we term them enteroendocrine cells. We confirmed that we could identify clusters enriched with expression of these different hormones, and also that, as compared to normal pancreas, we could identify solitary cells in metaplastic ducts expressing each hormone. We also performed transmission electron microscopy, and we could identify solitary cells with granules consistent with serotonin or somatostatin expression. Now, if you're a basic biologist or you're familiar with developmental biology, and Farzad gave a really good introduction to this in his talk, you're familiar with the concept that transcription factors are required for different lineages to form. For example, PTF1A is required for acinar cells to form. If you delete it in mice, you do not form acinar cells. NGN3 or NUROG3 is required for the formation of the islet lineage. And then within those cells, different transcription factors like PAX4 and ARX are required for each of these individual cell types to form and for them to express hormones. So we performed Regulon analysis on our data set, which looks at expression of both the transcription factor and all of its predicted downstream genes so that we can determine whether or not that transcription factor is on or off. And we found here that many of the transcription factors required for enteroendocrine and islet cells to form are expressed in this lineage. When we look more closely at some of these subtypes, like the enterochromaffin cells, we see that FEV, which is a transcription factor more recently published to be required for this lineage, is expressed. So what we predict that we're seeing is this concerted program where these transcription factor cascades are occurring, and it's required for these different cell types to form. Most of these mouse models are available, so we can generate a lot of different testable hypotheses here. But the big point that I want to make is that, consistent with our tough cell data, we predict that these are functional cell types, and they're using the same exact pathways to form as you would see in a normal organ. These cell types form in response to injury in human ADM as well. So again, this is a single nucleus RNA sequencing study with a few pancreatitis patients. We can see tough cells, mucinous ductal cells, endocrine cells. And then as compared to near normal in the pancreas, we can identify solitary cells in the metaplastic ducts expressing all 4 of the hormones that I mentioned. Now, do these populations populate precancer and PDAC? So to answer this question, we aligned our data set with a single-cell RNA sequencing data set generated by Schlesinger et al., which includes PANIT and pancreatic cancer. And you can see we still have this EEC enteroendocrine cell cluster present. We performed a histopathological study, which we published several years ago, where we looked at the pancreata of mice with either a KRAS mutation or a KRAS and P53 mutation. We stained here for synaptophysin, which is a general endocrine marker. And then we quantified the number of positive cells in each lesion. We did hundreds of lesions. And as you can see here, enteroendocrine cell formation appears to be an early event in disease progression, consistent with these cells forming in metaplasia. They make up about 10% of these ducts, so it's not a minor amount of cells. And we did the same thing for all 4 of the hormones that we identified in our injury study. So consistent with the general marker, we saw that somatostatin and pancreatic polypeptide are most abundant in metaplasia and in general decrease with disease progression. And the same thing here for both serotonin and ghrelin, where they're both most prevalent in metaplasia and then lost with disease progression. So we looked at abundance in human disease as well. So here we looked at 11 PDAC patients, but multiple sections so that we could look at normal tissue, early Pannin, and cancer. We stained for the same 4 hormones. And as you can see in the immunostaining, we can identify ghrelin, somatostatin, serotonin, and pancreatic polypeptide solitary cells, similar to enteroendocrine, integrated into metaplastic ducts and neoplasia. When we quantified these cells, we found that about a quarter of all ADM had at least one positive cell. This decreased a bit with disease progression. And then interestingly, I think, given the context of a lot of other people's talks today, we did see a bit of an uptick in pancreatic polypeptide cells associated with the cancer. This could be a response to the cancer. We can't do lineage tracing in humans, so this could be broken up islets as well. But the fact that these cells are integrated into the lesions suggests that they have the potential to communicate with cells in the ducts. And so this has been largely a marker study and pathology study, but a lot of questions come out of this, including, can these cells interact with islets? Somatostatin and pancreatic polypeptide could influence beta cell activity. There are other studies that suggest that somatostatin could be anti-inflammatory and therefore signaling to immune cells, whereas serotonin could be pro-inflammatory. And again, since most of us have metaplasia and Pannin, could the composition of those Pannin in terms of enteroendocrine cell abundance and subtype control disease progression? So these are a lot of future directions that we are interested in. To very quickly discuss what I think is a very clinically relevant phenomenon that we're seeing is that these cell types also express both incretins and incretin receptors. And so as many of you know, these incretin receptors are the target of many of these really popular weight loss drugs right now, including semaglutide, Ozempic, tirzepatide. And so what we're seeing in our metaplasia data is that these enteroendocrine cells express both GIP and the GIP receptor, which would be agonized by tirzepatide. So you can see here in our lineage tracing, we see GIP-positive cells derived from Asner cells, GIP receptor-positive cells derived from Asner cells. And when we stain different stages of disease, you can see it's present in normal islets. Again, they appear to be most abundant in metaplasia and decrease with disease progression. So it's not necessarily a question of whether people with pancreatic cancer should take these drugs, but if all of us have Pannin, how safe is it to take these drugs? And then finally, we also see GLP-1 receptor very highly upregulated in metaplasia and neoplasia in the mice, shown here by single-cell RNA sequencing, but again, in our lineage tracing study, this is not specific to enteroendocrine cells. We see this more in our mucin ductal population. And again, we see massive upregulation in metaplasia and then neoplasia, but a loss in cancer. So this question of whether or not these drugs would affect disease progression is more, would they affect Pannin progression? We did look in human disease. This is a pancreas atlas that was generated by Dr. Nina Steele here. We looked for our markers. We removed islets because the expression of GLP-1 receptor, GIP receptor, is very high in islets, but you can see that consistent with previous studies. We see GLP-1 receptor in Asner cells. We also see it in ADM with what looks like smaller expression in Pannin, but we can identify GLP-1 receptor by RNA FISH and immunostaining in Pannin. And then we see pretty high expression of GIP receptor actually in Pannin, and even some in cancer, which needs to be validated. So in summary, what I've told you is that Asner cells are surprisingly plastic, consistent with Farzad's data, and they can transdifferentiate into a number of cell types that are not present in the normal pancreas, but form during disease and can be found in normal glands or the intestine or the biliary tract. And so what we're seeing is this concerted program of gland formation, which appears to result in the number of functional cell types that can mitigate inflammation and disease progression. The enteroendocrine cells that form are heterogeneous. We've seen that a lot of these hormones don't overlap in expression, so we're seeing distinct cell types form, and they likely depend on the activity of specific transcription factors to form. The there's a lot of questions that come out of this. Could enteroendocrine cells impact islet cell function? Would they be pro or anti-diabetes in the context of chronic pancreatitis or PDAC? As many of you know, once the diseases form, there's actually a massive loss of normal tissue and metaplasia. Could the loss of metaplasia in these cell types also be a big driver of disease progression? And then finally, a question for thought, should ADM and neoplasia be something, in theory, considered when taking these medications? Because these could be considered to be normal cell types if all adults have them. So with that, I'd like to thank my laboratory, our collaborators at VUMC, the University of Montreal, and our funding sources, and the metaplasia GLP-1 receptor experiments are being supported by the Vanderbilt Diabetes Research Training Center. Thank you.
I'd like to invite all our speakers to come and join us here, and I'll open the room for any questions you have. I'd like to quickly start with a question. I was thinking it's very interesting, this finding about the insulin and the glucagon that we see in the diabetes in patients with pancreatic cancer. I'm wondering if there would be a role of using this in patients with new onset diabetes to help us, for screening of potentially for pancreatic cancer.
That's a really good point. We should, I think we could talk to Suresh Chari, who would be responsible for the actual biobanks where we could measure those things. Yes, good point. Suresh, where are you? Something to do. Oh, okay.
So we'll bring it up to him later
We'll tell him.
Please introduce yourself and where you're from.
Okay, sure. Suhail Hussain from Stanford. Really fascinating series of talks. I was particularly struck by Farzad's talk, and Farzad, I'm missing our interactions. We were one floor apart in Pittsburgh. I could almost hear your experiments from the floor below. But the question I have is that, it's so fascinating that you discovered that FAC, an inhibition or deletion is important for acinar to beta cell transdifferentiation. But I guess, and then based on either the spatial work that the periacinar tissue may be the nidus for this, should we be less stringent in our TPIAT islet isolations such that we capture more acinar tissue and somehow embed that in the liver, allow it to function in the way that you've expressed?
Well, I'm not a physician, so I cannot answer that question, but yes, theoretically, yes. Because, and the thing is that when we serendipitously, stumbled on this, because we handpicked the islets and we saw this, hybrid cell population or cluster. So if we had been stringent, and and many times you do that, got rid of all the acinar contamination, we would never have stumbled upon that. So maybe we could do that and include that. Maybe and if the signals are there or whatever, maybe they can transdifferentiate. Yes. In practice, I don't know whether that's possible or not.
Hi, this is Qian from the affiliated hospital of Qingdao University. And first, I want to thank you all for your amazing lectures and topics. And I have one specific question for Professor Pendle. So especially regarding the functional role of islet cells in the pancreatic cancer microenvironment. So during my PhD study, I studied fatty liver disease, and my current research focused on fatty liver and pancreatic cancer liver metastasis. And I noticed there's a slide showing that the specific knockout of islet beta cells, right? And this knockout of beta cells actually inhibit liver metastasis in a metastasis model. And this is amazing since islet cells in the pancreas actually can affect liver metabolism. And I believe that's most happen in hepatocytes. So how does this communication between islet cells and hepatocytes, and is it through like exosomes or any mechanism you can?
Your point is extremely interesting to me. So we actually, when we study the liver, when we change the beta cell expression of CHOP, like you said, we actually see changes in metabolic pathways in the liver. I don't have the answer right now, but it shows that the cancer is influencing a metabolic process that involves the liver that can affect the outcome. Extremely interesting, and these days I think that pancreas cancer is a metabolic disease, and we have to consider what you're saying and what we're hearing today and the understanding of the promotion of the disease. I do believe that the liver and potentially adipose tissue that we've heard about also are really important in the pathogenesis, yes. We don't have the answer yet, so keep thinking.
Okay, thank you. Thank you for that.
Andy Gunderson, Ohio State University. My question's for Farzad. So FAK inhibitors are being trialed in pancreatic cancer, mostly due to their blockade of stromal inflammation. So do you think in your studies the resolution of insulin resistance is due to inhibition of macrophage inflammation or fibroblast activity, et cetera?
That's a work in progress. So we don't know exactly the mechanism, what's going on. So we know that, of course, it inhibits, phosphorylation of FAK. So that's all we know. But the downstream targets, we have some ideas. I cannot disclose them right now, of course, but unfortunately, but we're getting there.
Sorry for the question. Okay.
Yeah.
Thank you.
But it's not macrophages.
So I'm here from University of Miami. Dr. Pendall, I have a question for you. So there was a recent phase 2 clinical trial by the Hopkins group, Dr. Yaffe, I don't know if she's in the audience, but so basically CXCR4 antagonism showed, number one, that you're seeing more infiltration of the immune cell, yes. It was interesting when you couple it with anti-PD-1 therapy, you're seeing, great response. But the downside was that the myeloid cells, the CD20 positive myeloid cells, so they were causing all immune suppression. Considering that in your research, like did you see, first of all, such a suppression with coupling those therapy? Well, you didn't do that therapy, but I'm asking from your experience if you can comment on that. And in future, like, are you thinking about using the XBP1 inhibitor in that sort?
Right. Also an excellent point. So actually, I'm asking my colleagues involved in CXCR4 inhibitory studies to look at diabetes, because I'm not sure that was addressed, because I was focused on that. But you're right, none of these things are simple because there's other cells we could affect. So we have to find the signals that may be, beneficial across the board, let's say across different cell types. But your point is excellent. That I was focusing on diabetes, but there's other effects of agents. So I need to, with my colleagues, look more carefully at the data from those studies and see if there's something there. But thank you for reminding me.
Thank you so much, sir.
Hi, Mark Wiley from UCSD. And I had a question for Dr. Del Giorno. Really fascinating talk. I was really interested in the enteroendocrine cell story. And I was curious how far you've taken that. I see the receptors and everything on there, but are these cells truly sensing and responding to nutrients in the pancreas? And how far have you taken some of that RNA-seq analysis? Like, are you able to see like L cells versus K cells within that population? And really what your thoughts are on what sensing and responding to those nutrients might do to that microenvironment.
No, that's a fantastic question, and it's still kind of a next step for us. So we've gone through the transcriptomic data pretty thoroughly, and we do see somatostatin receptors. So we see that these populations are present. We can kind of divide them into different cell types, like you're mentioning. As far as kind of K and L cells, we see the hormones, but by then you're starting to get into either really, really small populations, or what also happens in the intestine is that some of these populations will also express multiple hormones, and I think that's more like what we're seeing, like ghrelin positive and GIP positive cells. We see that our enterochromaffin cells also express gastrin, so a little more similar to the gut. That's a fantastic next step. As you know, the pancreas is kind of a pain, so we've been thinking more about ways to do that, because they do not automatically form in organoids. You lose them as soon as you make an organoid because they're very dedifferentiated. So we've been talking to more people about potentially looking at slice culture. That might be the best way to act, and that's something that your lab does as well. but that's a way to keep the cells intact and in their natural environment, and then to add hormones, look for calcium signaling, and then look for secretion.
Thank you.
But thank you.
Hi, Brittany Bergeman. I'm a pediatric endocrinologist and type 1 diabetes physician scientist at the University of Florida. My question's for Dr. Esney. Our group has also found these amylase negative clusters around the islets, which is really interesting, and we actually found that there are fewer of them in type 1 diabetes, so maybe a future direction for your group. But my question is more related to these beta cell acinar hybrids and how you found more conversion to beta cells in older mice. Have you all also looked at the NOD mouse or potentially NPOD, type 1 diabetes human tissues or anything like that?
So I have to disclose that we have encountered a huge amount of skepticism from the field. Of course, it took us 10 years to publish it. So that's a sort of in context of that. So we have tried it in NOD mice. Going back to, so we treated the NOD mice with the drug, and we saw improvement temporarily. And then, they were back. So that means that, so these are very, very much like normal beta cells, fortunately, but unfortunately they are hit by the, so we're still working on that. So that's the thing. So we will see, we still have to maybe suppress, do the immune suppression, but then, we get the conversion. In the older what was the question actually?
I was curious, I think I got this right, but your slide was mentioning there was more like natural conversion of these hybrid cells to beta cells in older mice. I was wondering if you also saw that conversion in an NOD mouse or anything with type 1.
So we haven't seen it in NOD mice because we cannot do the lineage tracing. So that's the only NOD data that we have. It's treatment, but normally we have seen it in normal sort of ELA tomato mice, lineage traced mice. In humans, I have a grant in pending. So whenever the shutdown is over, hopefully you get the something. I don't know.
If I was a reviewer, I would approve it. Thank you.
Our last question.
Hello. Great talks. My name is Maria Fields. I'm from Cincinnati Children's, and I am an assistant professor, and I am an immunologist. I have a question for Dr. Del Giorno. I was curious to see if you look at any of pro-inflammatory cytokines secreted by the tough cells, since in the intestine they've been shown to produce cytokines to skew on the immune system towards a Th2 phenotype and maybe promote fibrosis, or if you have looked at interactions between these cells and immune cells.
So we have done that. And it's interesting because the tough cells in the injury model reflect more the transcriptomic file that we see in the intestine, and the KRAS don't. So the injury express IL-25 and the KRAS don't. One thing that all the tough cells have in common is that they produce eicosanoids, which is a pathway that's just a little more difficult to study because they're lipid signals, but they express COX-1 and they express HPGDS. So in addition to IL-25, they'll secrete prostaglandin D2, which also induces a type 2 immune response. So at least in our KRAS model, what we've seen is that if we delete tough cells or prostaglandin D2, that we get more inflammation and more fibrosis. The tissue will eventually calm down, but it takes longer. So by the time it calms down, we have more disease. In the injury model, which is a project we're trying to get funded, but if we delete either tough cells or IL-25, again, we get more stroma, delayed edema, and more CD8 positive T cells.
Thank you.
All right, thank you so much, everybody, for your kind attention. I'd like to thank my co-moderator and the wonderful speaker for this very engaging session. Now we're lunch.